Hello, and welcome to this Ethical Dilemma in Science, where we're going to look at future options for organ transplants. And I'm describing this as making a silk purse out of a sow's ear. Basically, what we're going to look at is this idea that about 17 people a day, or at least it's estimated, to die each day waiting for an organ transplant. And if we take a look at what is kind of known about the statistics, and these are based on 2020 statistics, so available in the beginning of 2021, they're showing that about 90% of U.S. adults support organ donations, but we have an opt-in system in the United States. And so in order to be an organ donor, you need to sign up. And so even though 9 out of 10 U.S. adults, American adults, support organ donation, only about 6 out of 10 are actually going to sign up as organ donors. And then, see, we've lost, a large population right there. But then we take a look at less than 1%. Basically 3 out of 1,000 people, 0.3%, die in a way that allows for organ donation. And so again, we can see what is going on that makes it very, very difficult to be able to get these organs to the patients that need it. Now, as we've talked about, it's going to be very important that the donor and the recipient are going to be matched to minimize the risk of rejection. And so if we take a look at this, and again, this is similar to blood typing, and I use this example because blood typing is a little bit easier to explain than organ matching and the concepts associated with it. But the same basic idea is there. It's just using different kind of components within the cell. Basically, there are going to be components on all of our cells, little structures. There are going to be little proteins, but you can think about it as little shapes. They're going to be on the surface of your cell, and they're going to be different from one individual to others. But they're not totally random. They're not totally unique. They're going to be categories. And so when we talk about blood types, you've probably heard that you're either going to be type A, type B, type AB, or type O blood. And so what that means is individuals that are type A are going to have this A structure, kind of a little purple lollipop structure on here. And the scientific name for that is an antigen, but it's basically a structure, a protein that's on the surface of the cell that has a unique shape associated with it. And then individuals with type B blood are going to have little green, little diamond-shaped structures like we've got over here. And so basically that means that individuals with type B blood are going to have different proteins on the surface of their blood cells, and they're going to have different shapes on the surface of their blood cells. Individuals with type AB blood are going to have both the A shapes, the purple lollipops, and the B shapes, the green diamonds, on the surface of their cells. And then people with type O blood are going to have none of those. Their cells are going to be kind of bare. They're not going to have these shapes. There are other things there, but at least the proteins we're looking at, the shapes that we're looking at, aren't going to be present there. Okay, so that's interesting, but what does that have to do with matching a donor to a recipient? What we have within our bodies is an immune system. And as we're coming out of the COVID-19 pandemic, people have thought about their immune system a lot. Basically, what happens within your immune system is that you're going to have cells that are going around throughout the body looking at either invading cells, cells that have come in from the outside that, are different from the normal body cells, or disease cells. And so what happens then is that these invading cells, potentially pathogenic cells, disease-causing cells, or cells that have been damaged in some way and have a disease process like cancer associated with them, they are going to have shapes on the surface of their cells that can be used to recognize them as being abnormal to the body of the individual we're looking at. And the way that they're going to do that is they're going to be producing antibodies. And again, lots of talk about antibodies and sera and things like that, blood components over the last year or so. But basically, these are little flags that are going to go through and bind to the shapes of these things that are on the surface of the cells. And so individuals with type A blood are going to have the little purple lollipops associated with them, these rounded shapes. So what they're going to have are going to be anti-B antibodies. That's a fancy and complicated way of saying that they are going to basically recognize these green diamonds as being foreign. These green diamonds are going to be recognized as not normally within the body. They're probably not going to be good for the body, and so they need to be eliminated. And so these antibodies are like little construction flags. They're going to bind to the B-shaped, these B-shaped green diamonds here. And they're basically going to say, this is bad. This isn't normally here. We need to get rid of it. And other cells come along and kind of destroy it. And so what we see with blood donations, if you give type A blood to a type A recipient, they both got these these purple lollipops on their surface, and so everything's fine. And we've got a good match here, and the blood is able to go into that recipient, and everybody's happy. The problem is if you take type B blood, they're going to have the green diamonds on their surface, those B antigens. And so when that B blood gets into the body of an A recipient, their body, specialized cells within their body are going to say, hey, we haven't seen this before, and this is probably not good. We need to get rid of it. And so basically the antibodies are going to bind to it. It's going to cause the blood cells to clump up and actually be destroyed. And so this is kind of not good. And so this would be the equivalent of a mismatch between a donor and a recipient. And so in this case, we would have the situation where if this was an organ, the recipient's body would be actively trying to destroy the cells because they would recognize them as being foreign, as being diseased, as being potential trouble associated with it. And so what that means then is that what we'd look at in matching between blood donors and blood recipients is we're looking at these molecules along the surface and saying, okay, is there a situation where we can take the blood from this individual over here, from this donor, and put it in a recipient, and that blood is not going to be recognized as a risk. And if it's not recognized as a risk, it's not going to be rejected. And so that is what's occurring within blood donation. And we can see that type O blood with none of these proteins on it, none of those shapes associated with it, can basically donate blood to all individuals. But an individual with type O blood here is basically have antibodies. They're going to recognize the A purple lollipops in the green, the B green diamonds as being foreign, and they're going to eliminate them. And so they're not going to accept type A blood, they're not going to accept type B blood, and they're not going to accept type AB blood, which actually has both of the shapes associated with it. And so what we're looking at when we're talking about organ and tissue rejection is we're doing the same basic thing with other types of molecules that do the same kind of basic thing as these kind of purple lollipops and green diamonds up here. Even with the best match of organs going from a donor to a recipient, we are generally going to be treating those patients with immunosuppressants to kind of minimize their immune response, which is good for the survival of the donated organ, but it also means that they're at higher risk of getting infection. But we need to have organs. We know that there is this dire need for transplant organs. We talked about the statistics, 17 people dying every day waiting on the list. And so there have been efforts to try to find, are there other avenues of getting these cells? And so one idea that was looked at was cross-species organ transplantation. And so in this case, we're looking at a man in the early 80s that got a baboon bone marrow transplant because his blood cells were basically dying. We had baby Faye about the same period of time that lived about 30 days with a baboon heart. But basically these are situations where it's the last case. It's the last option available to them. And so they've tried different organ transplants from other species and it hasn't worked out as well as we'd like. Still, there are ways in which we can take organs or structures from other species. This is a pig valve right here, a heart valve that has been isolated from a dead pig that can be used for transplantation into humans. If we take a look at the two species that are most commonly used for cross-species organ transplants, we've got primates and we've got pigs. Primates were thought about as an option because there's less risk of rejection because they're closer to the human characteristics, the human kind of shapes that are present within their cells. But there's a higher risk for diseases that can be passed on the donor tissue, and there's a relatively long generation time. It takes a long period of time for the primates to grow to a size where the organs can be transplanted. Pigs, on the other hand, have less risk of transferring disease because pigs have been domesticated and have been close to humans for a long period of time. They have a relatively short generation time, so you can go from kind of a newborn pig to a 250-pound full-size pig in a little less than a year, and so their body size is comparable to those of a human being. Now, the problem with pigs is that they are very, very different. And because they're very, very different, they're going to be more likely to be recognized by the human recipient's immune system as being foreign and being rejected because of the shapes along their cells. And so what we look at is this idea, and we'll talk about genetic engineering later on within this course, is genetic engineering kind of addressed one of these problems. Patients with diabetes are often treated with insulin. And so insulin was first isolated in 1922, so roughly 100 years ago. And for a long period of time, diabetes was treated with either pig or cow insulin. And that pig and cow insulin had a shape that was associated with either a pig or a cow. It still had the function of insulin, but there were individuals that often mounted an immune response to the pig insulin or the cow insulin, isolated from the organs of either a pig or the cow. And so because their immune system responded to it, they would reject the insulin. They would not be able to use that, and therefore they would lose out on the most common treatment for diabetes. Within the 1970s and in the early 80s, they used the concept of genetic engineering to take the gene for human insulin, take the information for making a human insulin molecule, and they put it into a bacterial cell, and they had those bacterial cells producing human insulin. And so they're basically able to produce what would be needed by these patients that are mounting the immune response against the pig insulin or the cow insulin. They're able to get human insulin without having to try to figure out how are we going to get human pancreas, that's the organ that produces insulin, how are we going to get those and grind them up. Huge ethical issues kind of considering that. And so, again, relatively recently, within the last year or so, we've seen that pigs have been genetically modified to be less allergenic. They've basically gone through and change the molecules that are present on the surface of these pig cells. And so going back to our blood donor example over here, this is equivalent of taking these pigs that had these shapes on their cells and getting rid of those shapes. And this is dramatically oversimplified, but I'm trying to illustrate what is potentially coming down the road. With this idea that they don't have these shapes on their surfaces, they can then be donated to any of the recipients, because without those shapes, there's nothing for these cells to be recognized as being foreign. This is so significant that the Food and Drug Administration of the United States, within the last year, December 2020, came through and approved these for both human food and potential therapeutic uses. And so they recognize them as effectively safe, and then opening up the avenue for lots of things that we can be looking at further on. In terms of ethical dilemmas associated with it, again, starting out with this basic concept of xenotransplants. A xenotransplant would be a transplant from one species to another species. Right now, recipients of xenotransplants are required to participate in long-term monitoring because of the risk of an endogenous zoonotic disease, a disease that could come over in the tissues from the donor and then become established in a human. And in terms of zoonotic viruses, we've got a pretty good idea about what one of those can do with COVID-19. So there are huge theoretical public risks associated with it. Should these recipients be required to participate in this monitoring? Should recipients of the xenotransplants be required to inform close or intimate contacts of potential health risks? This was something that occurred in the early 1980s and 90s, where there was a requirement for patients with HIV to let their partners know that they're at risk of transmission. In terms of taking a look at these pigs, what is the ethical dilemma associated with this? Should scientists continue to alter the pig's DNA to make it less pig-like, to kind of eliminate those shapes along the surface of the pig to make them more like, or at least more acceptable to humans and minimize the risk of rejection? The flip side of that is that, if we're making these cells less pig-like, we have the potential to modify them to become more human-like. And so this would be like taking the gene for insulin, the information for building insulin, and putting it into a bacterial cell to get the bacterial cell to produce insulin for us, to produce human insulin. Should scientists begin to modify the pig DNA so that it becomes more human-like? And what limits should there be for determining how human-like should these cells be before there are limits put in place? And then finally, again, this idea of equity and availability of resources in a fair and equitable way. We have the potential to look at designer pigs. Should there be strains of pigs that are created to meet the needs of broad tissue matching characteristics? I oversimplified it a lot to say type A, type B, type A, B, and O, but there are certain molecules that are present on the surfaces of cells, and if we have those, then they could be available to individuals that have those proteins, have those shapes on the surface of their cells. Should we take this a step further? And, a wealthy individual, can they create, or order a specially created pig that matches their own unique characteristics? So they've got their own little tissue bank on a farm someplace that is growing and producing organs that they can use for in their body. And then finally, this idea of chimeras. Chimeras in mythology were creatures that were put together with different species. Should there be a limit on how much of a body can be replaced with these spare parts? Should it be permitted to prolong life beyond what we would consider the traditional lifespan with these spare parts? And then an area that is of concern right now is, will there be emotional or societal impacts on the recipients that have had their lives prolonged with these donor animal organs? We're not talking about, a true, pig boy type situation, but we're looking at kind of a mixing and matching of human and non-human parts to come together. And so what are the implications of that? So thank you for viewing this ethical dilemma. Please let me know if you have any questions. Thank you and have a great day.